Molecular basis of inheritance, biology part 2, speed run. I'm going to continue it right now. I'm going to continue from the topic I left off yesterday, which I didn't do yesterday. Basically genetic code, which I'm going to start today. Genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Okay, during replication also and transcription also, a nucleic acid, which can be DNA or RNA, DNA in case of replication and RNA in case of transcription. Wait, no. Both DNA in case of both replication and transcription was copied to form another nucleic acid, which is DNA in case of replication and RNA in case of transcription. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic material from a polymer of nucleotides to synthesize a polymer of amino acids. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acid. Basically, translation itself, we can understand they are translating the code. They are translating the code into a protein. The code is nothing but the nitrogenous basis code, which will be NATTG, whatever it might be. That is, they are converting the code into amino acids. That is how DNA actually is actually the blueprint that is actually storing all the information. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. Basically, we know that RNA and DNA or DNA are complementary, because there will be exact opposite codes in the, basically if one we know, other we can find out, even for RNA or DNA. But there is no complementarity between nucleotides, basically adenosine, guanosine, cytosine, uracil and thymine, between these and amino acid, lysine, arginine, valine, glutamine, etc. These are all amino acids which form proteins, and nucleotides which form nucleic acids. There is no complementarity between them. There existed ample evidences, though, to support the notion that change in nucleic acid genetic material were responsible for change in amino acids in proteins. Basically, there existed in the olden times evidence, even now it's there, even it is more thorough right now. When they didn't completely know what was going on, there existed evidence that supported the feature that, or explained that nucleic acids were responsible for change in amino acids in protein. The nucleic acid changed the amino acids in the protein, changing the protein itself. This led to proposition of a genetic code that could direct the sequence of an amino acid during synthesis of protein. Basically, if nucleic acid is producing or changing the composition of the proteins, how is it doing it? So in that idea itself, this led to a proposition, that assumption led to a proposition of a genetic code, a code that could direct, meaning that could assist or perform or help in the sequence of or formation of proteins, that is arranging the sequence of amino acids. There are 20 types of amino acids, and different combinations of these amino acids in thousands of monomers formed together will form a single protein. If determining the biochemical nature of a genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were the most challenging. Determining the biological nature of a genetic material and the structure of DNA was very exciting. Basically, what is the genetic code? We saw in the Griffith experiment that there was a transforming principle, and Hershey-Chase experiment that there was basically basically genetic DNA is a genetic code. That was one of the experiment. And the structure of DNA, how structure of DNA will be, how transcription happens. This is also very exciting, very interesting. But the proposition and deciphering, understanding or knowing genetic code was most challenging. In a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. It was many of these people, even physicists, organic chemists, biochemists, and geneticists, all these people were involved for finding and deciphering the genetic code of the DNA. It was George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. There are only 4 kinds of bases, but there are 20 kinds of amino acids. And if proteins, if we see, there are many types of proteins. So this physicist George Gamow thought that because of such a reason, then how can 4 bases code for 20 amino acids? That is not possible. So he came up that there should be a combination of codes, not a single code, a single base coding for a single amino acid. If that was the case, only 4 amino acids 4 bases can code for. So that is not the case. So he thought that a combination of these bases will be formed, and these combinations will lead to the formation of this 20 amino acid, constitute a code combination of the bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. Basically, in order to code for all the 20 amino acid, the code should be made up of a 3 nucleotide. Basically, the code should be made up of 3 nucleotides. This was a very bold proposition because our permutation combination of 4 power 3, which is generating 64 codons, generating many more codons than required. There are 20 amino acids. Codons is almost more than 3 times the number of amino acids, which is much more than what we required. We required only for 20 amino acids, but he's telling that we need 64 codons. So that was very high, generating many more codons than required. And this is actually what's correct here, and it was a physicist who actually came up with this idea, providing proof that the codon was a triplet was more daunting task. Basically, because 3 amino acids, so basically 3, and this 3 can be any 4. So basically, using the formula here, we'll get 4 times 3, 4x 4 times 4 times 4, which will generate 64 codons. Because there are 3 here, we call it a triplet codon. And how should we prove that codon is actually a triplet? That was very hard. The chemical method developed by Hargobind Khorana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers, copolymers, Marshall-Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Let's read that again. Basically, the chemical method, the first time 2 scientists are introduced here. That is Hargobind Khorana, and Marshall-Nirenberg. Hargobind Khorana and Marshall-Nirenberg. These 2 are the scientists, and Hargobind Khorana was discovered or developed a chemical method instrumental in synthesizing RNA molecule with defined combination of bases, homo or copolymers. Basically, Hargobind Khorana, he developed a chemical method from which we can instrumentally synthesize RNA molecule with only a specific or a defined combination of bases. It might be AGT, TCG, or any kind of certain. We know the code. Basically, we know what kind of code is there. We will know that code only will be produced. He developed a chemical method in where we can do such a thing. And the next, Marshall Nirenberg's cell-free system. Basically, he developed a cell-free system, meaning proteins can normally only be synthesized in cells. But Marshall Nirenberg prepared a method we can prepare proteins outside the cells as well. And these two, a chemical method to create RNA polymers, RNA molecules with defined combination, and Marshall Nirenberg's cell-free system for protein synthesis, finally helped the code, the genetic code, to be deciphered. Severo-Ochoa enzyme. Severo-Ochoa enzyme. How to pronounce?
Ochoa.
Ochoa. It seems Severo Ochoa enzyme, polynucleotide phosphorylase. Basically, it is a polynucleotide, many nucleotides, phosphorylase. And this was an enzyme that was helpful in polymerizing RNA with defined sequence in a template independent manner. Enzymatic synthesis of RNA. Normally, we require a template for knowing the exact for getting, actually, for in case of transcription, we saw mRNA was formed through a template being the 5 prime to 3 prime. So yes, 3 prime to 5 prime was a strand that was acting as a synthesis, that was acting as a template. But here in this case, there was an enzyme, Severo-Ochoa enzyme, which is a polynucleotide phosphorylase, which was helpful in polymerizing RNA with defined sequences. We know the sequences in a template independent, not in a template manner. This is called the enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in table 5.1. The codons for various amino acids. This is the first position. This is the third position. Basically, the first is U, which is uracil. We take uracil and not thymine here because mRNA is the one that, in case of mRNA is the one that will code for that is getting translated. mRNA is getting translated, so not the DNA. So we need to take uracil itself here. U is uracil, C is cytosine, A is adenine, G is guanine. So basically the checkerboard is like this, and U, basically 3 positions, one here, this U, and this U, that is one, and this U, and this C, A, UUG. Basically triple U codes for phenylalanine. No, wait, I don't know. PHE, basically that is also an amino acids. PHE, LEU, ILE, MET is methionine, and VAL is valine. TYR, I think, is tyrosine. ARG is arginine. and there are specific stop codons here. And if we see, this is the table, this is basically a checkerboard table where we see all kinds of amino acids coded for here basically. So this is the first table. This first table is taking this one and this one as constant and gliding through this table. In case of here, taking this one and this one constant, again gliding through this values. Here taking this one and this one common, the third table, and gliding to this table. The same way it is done. And there are special codons as well. If we see, these are all mostly all are if we count 16, 32, 46, basically out of the 64, 61 are actually only amino acids. 64 are amino acids. 61 are amino acid codons, not amino acids. 61 are amino acid codons. If we see here, there are 3 codons, which is UAA, UAG, and UGA. These 3 codons are for stop codons, ending the translation process. And there is also, if we see, there is a unique amino acid here that is only coded by one of the codons, that is methionine, AUG methionine. Here if we see AUG, there is no methionine itself. Only AUG can code for methionine. And this AUG is called the start codon. It is the first codon in any kind of protein. Start codon. And these 3, AUG, August, AUG is the start codon, UAA, UAG, UGA are all stop codons that will end the translation process. Now let's see. The salient features of genetic code are as follows. The codon is a triplet, as we saw here. We take 3 bases here. 61 codons code for amino acids and 3 codons do not code for amino acids, and they function as stop codons. They do not code for amino acids, so they will stop the translation process. Some amino acids are coded by more than one codon, hence the code is degenerate. Okay, basically if we see here, if we see valine, if we see all these G1s, GUU, GUC, GUA and GUG all code for the valine amino acid. And in case of lysine here, AAA, AAG both code for lysine itself. And arginine if we see, CGU, CGC, CGA, CGG all code for arginine itself. So we can say the code is degenerate. The codon is read in mRNA is in a contiguous fashion. There are no punctuations. The codon is read in mRNA in a contiguous manner or fashion. There are no punctuation marks. This code is universal. For example, basically what they mean universal here means it is not only for humans, not only for mammals, not only for animals. In any kind of living organism, the code is same. This GGG compound here, GGG codon, triple G codon will code for glycine itself. For example, from bacteria to human, triple U would code for phenylalanine. Some exceptions to this rule have been found in mitochondrial codons and in some protozoan. Basically, as I said here before, the triple U and triple G will code for glycine and triple U will code for always phenylalanine only. But there are some exceptions. Mostly it will work like that only. But there are some exceptions. In such, where we can find this exception is in mitochondrial codons. Mitochondria has its own DNA and it also has its own ribosomes as well, and it can undergo self-replication. In such a case, in mitochondrial codons and in some protozoans, we will see such cases. AUG has dual function. AUG, this is the methionine. As I said, it's a start codon. It can act as a amino acid, a normal amino acid, and it can also act as a start codon. It codes for methionine and it also acts as initiator codon. UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotide of mRNA, predict the sequence of amino acid coded by it. Take help of the checkerboard. AUG is the initiator codon, methionine. Triple U, it is, I remember it was phenylalanine. UUC is also phenylalanine. Methionine, phenylalanine, phenylalanine, again phenylalanine, 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 and again phenylalanine. All are phenylalanine itself. Methionine started with a code for phenylalanine. And next is, now try opposite. Following is a sequence of amino acid coded by an mRNA. Predict the nucleotide sequence in the mRNA. Methionine, phenyl, 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 alanine, alanine. Did you face difficulty in predicting the opposite? Yes, of course we will face difficulty, because if we see phenylalanine, we know that methionine is AUG. But what about phenylalanine? Phenylalanine, we have two codons here basically. Two codons, triple U and WC. In such a case, which we should take as WC and which we should take as triple U? We don't know, right? Can you now correlate with two properties of genetic code? We have learned two properties. I'm not too sure what that means, but fine. Let's continue. Mutations and genetic code. Can you now correlate to which two properties of genetic code? You have learned it is basically genetic code properties. It codes for coding, it helps in the translation process, and we can find out the amino acid coded for through the main genetic code, but we cannot find the genetic code through the amino acid itself. Okay, fine. Mutations and genetic code. The relationship between genes and DNA are best understood by mutation studies. Basically, they are saying the relationship between genes and DNA are best understood by mutation studies. Mutation is basically the alteration in the gene coding that will lead to changes. Yes, this we studied in the previous chapter. Studied about mutation and its effect in chapter 4. Effects of large deletions and rearrangements in a segment DNA are easy to comprehend. Basically, effects of large, very large number of codons base pairs if we remove, and rearrangements, large arrangements in a segment of DNA, they are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutations will be explained here. A classical example of point mutation, the first, basically it may, like if large amount of DNA segment is lost, it may result in a loss of a gene, but if it is added, then it might result in the gain of a gene, and both ways we will get a new function, a new character that is going to be expressed. And now the next topic about the main topic, not main topic, one of the mutation is point mutation here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue to valine. It results into disease condition called sickle cell anemia. Basically, a classical example for point mutation is a change in a single base pair. Only a single base pair is going to change for beta globin chain that results in the change of amino acid residue glutamine to valine. Basically, in case of normally the code will be there, and in this code there will be a change of a single base pair will be wrong, and in that case the amino acid will be there. Due to this case we should get glutamine here, but it will be replaced with valine. That will result in the disorder called, or disease called, sickle cell anemia. Sickle cell anemia happens because of the quality change in the globin chain production. Effect of point mutations that inserts or deletes a base in structural gene can be better understood by following a simple example. Basically, point mutations that can insert or delete a base in a structural gene can be better understood by following a simple example. Basically, consider a statement that is made up of the following words, each having 3 letters like genetic code. Ram has a red cap. If we insert letter B in between HAS and RED and rearrange the statement, it would be read as follows. Ram has BREDCAP. This is causing a change. Similarly, if we now insert 2 letters at the same place, like say BI, now it would be read as Ram has BIREDCAP. Now we insert 3 letters together, say big. The statement would be read as Ram has a big red cap. The same exercise can be repeated by deleting the letters R, E and D. One by one, the rearranging the statement would make a triplet word. Ram has a EDCAP, Ram has BCAP, and Ram has a cap. Ram has cap. And seeing this, if we see when we added a single base, it caused a chain reaction. If we see here, normally if it did not, when we added a single base here, it did not change the single base sequence, it changed the whole chain sequence. Here it should be cap, but it became DCA. And what about the next one? And here it should be red, but it became BIR, changing the whole sequence as we go to the next one. And in case of, but when we added a whole codon, which is BIG, it just added a single, like it only added a single extra amino acid. It only added a single extra amino acid, but in case of here, it changed the whole sequence itself. And in case of here as well, when we deleted 1 letter, it changed the whole sequence. 2 letters also changed the whole sequence. But we deleted 3 letters, it just removed an amino acid that was going to be in the protein chain. The conclusion from the above exercise is very obvious. Insertion or deletion of 1 or 2 bases change the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutation. Basically, when we added one or two bases, it changes the reading. This changes the reading of the base reading frame from the point of insertion or deletion. It changed the whole sequence itself. However, these mutations are referred to as frame shift because it is completely shifting the whole frame. They are called as frame shift insertion or deletion mutation. Insertion or deletion of three or its multiple bases, insert or delete in one or multiple codons, hence one or multiple amino acids, and reading frames return unaltered from the point onwards. Basically, insertion of deletion of three or its multiple bases, insert or delete in one or multiple codons result in one or more multiple amino acids, and reading frame remains unaltered from that point onwards. Basically, when we added three codons, three bases we added. Codon is a triplet, and codon meaning we are adding three bases. So when you add multiple codon or a single codon which is a multiple of three bases, so at that point a certain number of amino acids will be changed, but it will not completely shift the frame in case of frame shift or deletion mutations, and reading frame remains unaltered. The next concept is tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also link it to the amino acids. I think Francis Crick was the one who came up with the DNA double helix model. Double helical model. Yes, James Watson and Francis Crick came up with the double helical model based on the X-ray diffraction data produced by Morris Wilkins and Rosalind Franklin. Okay. Okay. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also link it to the amino acids, because amino acids have no structural specialties to read the code uniquely. Basically, when Francis Crick is the one who proposed the double helix model, and whenever this thing that there was a specific code that is arranging these amino acids to form proteins, whenever he heard this, he knew that there was a mechanism to read the code and also link it to the amino acids, because we know that amino acids do not have any structural specialties to read the code uniquely. He postulated the presence of an adapter molecule that would, one hand, read the code and other hand would bind the specific amino acids. He knew amino acids had no structural specialties to read the code uniquely. So he postulated, so basically he thought there must be a mechanism. So he thought that the mechanism was based on an adapter molecule, and the adapter molecule would have two functions. The first function is reading the code and bringing the specific amino acid, and the second function is binding the amino acid to the chain of amino acid. The tRNA, then called soluble RNA, sRNA, was known before the genetic code was postulated. Basically, the tRNA, transfer RNA, before was called the soluble RNA, sRNA, was known. We knew about this molecule before the tRNA itself, As we knew about DNA before we knew it was a genetic code itself. People knew DNA, people knew about DNAs, we knew about the molecules, but we didn't know genetic, what was the actual genetic material. Same way, we knew about the tRNA, which was called soluble RNA, and this was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. Basically, they knew about this, but they didn't know that it was actually the molecule that was assigned, that it was actually the adapter molecule, which was assigned much later. As we saw, DNA was invented as something present in the nucleus called nuclein, and it was in 1858 or something, but only in 1900s was actually found out that DNA is a genetic material. This is the adapter molecule tRNA. This is the tRNA structure. This is the amino acids at the 3' end and 5' end. 5' end is empty, and there is a linkage with the amino acids at the 3' end. This is the hydroxy end. Hydroxy end, there is forming a link, and here we see this is the mRNA from 5' to 3'. This is the UAC, which is coding if we do adenine to uracil and adenine to uracil here as well, guanine to cytosine. So this is exactly complementary. So this codon, this AUG codon, this is a start codon, will come and attach here. Wait, this is not the start codon. AUG is not the start codon. Wait, AUG. AUG. It is indeed the start codon, but I think they are considering it in base to the mRNA reference, where it should be, I think, like this. AUG. So basically the tRNA should contain, for A it is U, for U it is A, for G it is C. UAC is the start codon in reference to the transfer molecule. And UAC, if we see, UAC should be tyrosine here. UAC. UAC. See here, we got it as tyrosine. So basically all the codons are based on the mRNA sequence, not the tRNA sequence. tRNA has a loop, an anti-codon loop, that has bases complementary to the genetic code, and it also has an amino acid acceptor end to which it binds to amino acids. Basically, the tRNA molecule has two loops or end, one loop and an end. This is a loop, and this loop has bases, nitrogenous bases complementary to the code, and it also has an amino acid acceptor end. This is the amino acid acceptor end, which is a 3' end. tRNAs are specific for each amino acid. This AUG will only bind to UAC because of the complementariness. Figure 5.12. For initiation, there is another specific tRNA that is referred to as the initiator tRNA. There are no tRNAs for stop codons. Basically, they are saying tRNAs are only for initiator codons, which is a methionine. There are no tRNAs for stop codons. The secondary structure in the figure, the secondary structure of tRNA has depicted that looks like a clover leaf in actual structure. This is the clover leaf structure of the tRNA molecule, but in actual structure, the tRNA is a complex molecule which looks like an inverted L. Here it looks like a clover leaf, but in actuality it is a complex molecule that looks like an inverted L. Now let's see translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. This is one of the main topics here. Translation refers to the process of polymerization, binding together many amino acids to form a polymer chain of amino acids to form a polypeptide. Here each tRNA will bring the mRNA here, and this will go, and there will be bond formation between these two amino acids. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. This is what we have been reading for the past many topics. The amino acids are joined by a bond which is known as the peptide bond. Here I showed you a bond here, the amino acid bond. This is called the peptide bond. Formation of a peptide bond requires energy. Everything requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ACP and are linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be specific. Basically, this peptide bond formation requires energy. Therefore, this energy is provided not during the stage of bonding but before itself. In the first phase itself, amino acids are activated, provided energy in the presence of ATP, and they are provided energy and linked to their tRNA. Basically, tyrosine is linked for the AUG tRNA. It is linked here. Here itself, it's in the activated stage in the presence of ATP. If two such tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. If they are brought closer together, the bond formation is more favorable between amino acids than compared to tRNA. That is why chain bonding is formed. It is favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. If there was even a catalyst, it would increase the rate of bond formation, peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. This is the translation. This is the ribosome upper and lower units. This is the mRNA which is enclosed in the ribosome, and here the tRNA come with amino acid. Link happens because the peptide bond, they are activated, amino acylation, charging of tRNA already charged, and this bond formation is favorable. And once the tRNA used up, the tRNA will leave and the next tRNA will come. The ribosome consists of a structural RNAs and about 80 different proteins. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits. Basically, first information about ribosome is that it consists of structural RNAs. Okay, fine. And it consists also about 80 different proteins. That is the first information about 80 proteins and about how there are ribosomes, not ribosomes, RNAs inside the ribosome. And the next information is that in the inactive state of the ribosome, it exists as two subunits, a large subunit and a small subunit, which we know for the ATS is 40 and 60S. ATS for the ATS ribosome present in eukaryotes, it is divided into 60S and 40S subunits, whereas in 70S ribosome it is divided into 30S and 50S. I think that is right, yes. a larger and a smaller subunit. When the small subunit encounters an mRNA, when a small subunit encounters an mRNA, the process of translation of mRNA to protein begins. When a small subunit encounters an mRNA, basically meets the mRNA, the starting of the process, basically the process will start. There are two sites in the large subunit for subsequent amino acids to bind and thus be close enough to each other for formation of peptide bond. The next information is that the first information is about the small subunit, which when meeting the mRNA will begin the process of translation. And in case of the larger subunit, there are two sites in the large subunit for subsequent amino acids to bind, and thus basically amino acids should be close together for the energetically favorable peptide bond formation. They should be close enough. So that's why it provides space. This ribosome also acts as a catalyst. 23S RNA in bacteria is the enzyme of ribozyme. The ribosome also acts as a catalyst. Basically 23S rRNA, basically 23S rRNA in bacteria is the enzyme. This 23S rRNA is the enzyme, and it's called the ribozyme for the formation of peptide bond. This helps in the catalyzation. A translation unit in mRNA is a sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. Basically, a translational unit in mRNA is a sequence of RNA that is flanked by the start codon and the stop codon and for a polypeptide. A translation unit in mRNA is a sequence of RNA that is flanked by the start codon and the stop codon codes for a polypeptide. A translation unit in mRNA is a sequence of RNA that is flanked by the start codon to stop, and the stop codon codes for polypeptide. I'll be taking a 5-minute break. Okay, let's continue. Basically, a translation unit in mRNA, is a sequence of RNA that is flanked by the start and the stop codons and codes for a polypeptide. Basically, they're defining a translational unit here in terms of the codons. Basically, a translation unit consists of promoter, structural, and terminator genes, and they are, even though the promoter and terminator genes do not code for a polypeptide, they are called genes because they affect the process of transcription and translation. Actually, wait, no, this transcription unit is different. This is the translational unit. Okay. The translation unit also is a process with this is the in here also we can define a structure like the transcriptional unit. It was promoter, structural gene, and terminator. Here also it is mRNA, a start codon, and a stop codon, and this is coding for a polypeptide. This is basically a translational unit. That is what they're saying here. A translation unit has a start and a stop codons. I think the other one was transcriptional itself. Transcriptional unit. Yeah, it is a transcription unit. Fine. Let's continue. An mRNA also has some additional sequences that are not translated and are referred to as untranslated UTR regions. The UTRs are present at both the 5' before the start codon and 3' end after the stop codon. They are required for efficient translation process. Basically, before the start codon is attached, there is no translation process. These untranslated regions are present before the start and after the stop codon. In the middle is the one that is getting translated, but mRNA has places where these sequences, where extra sequences are there that do not have a start codon, and in such cases these are used for efficient translation process. For the translation process to be efficient, these are used. For initiation, the ribosome binds to mRNA at the start codon. Basically, the tRNA, not tRNA, the smaller subunit of ribosome binds to the mRNA at the start codon, that is AUG. That is recognized only by the initiator tRNA. Initiator tRNA, the methionine tRNA, AUG tRNA, the AUG is with respect to the mRNA. For initiation, the ribosome binds to mRNA at the start codon, that is recognized only by the initiated tRNA. The ribosome proceeds to the elongation phase of protein synthesis. Okay, then the ribosome proceeds to the elongation phase of the protein synthesis. During this stage, complexes composed of amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. Basically, for initiation, the ribosome binds to mRNA at the start codon. Okay, that is recognized only by the initiator tRNA. This binding site is only recognized by the initiator tRNA. The ribosome proceeds the elongation phase. Okay, elongation, continue the amino acid elongation of the protein synthesis. A ribosome, after the tRNA is connected, binding, Then it will start the elongation protein synthesis. During this stage, complexes composed of amino acid linked to tRNA sequentially bind to appropriate codon in mRNA, forming a complementary base pairs with a tRNA anticodon. Wait. Anticodon. Basically, elongation phase starts, and during this stage, complexes composed of amino acid linked to tRNA. During this stage of elongation phase, basically they are describing this process where one tRNA is coming here, joining, forming a peptide bond, and it's leaving. That is what they are describing here. During this stage, complexes composed of amino acid linked to tRNA sequentially, based on the sequence, bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA okay, this tRNA has a codon, right? This UAA in case of tRNA here, this is the anticodon. This is not a codon here. This AUG is an anticodon. The ribosome moves from codon to codon along the mRNA. Amino acid are added one by one, translated into a polypeptide sequences dictated by DNA and represented by mRNA at the end. A release factor binds to the stop codon. Basically, after the complete elongation phase, a release factor binds to the stop. Basically, there is a UAG, UAC, which is a stop codon present on the mRNA, and there is no anticodon for this. There is only a release factor which binds to the stop codon, terminating the translation, releasing the produced amino acid, terminating the translation and releasing the complete polypeptide from the ribosome. Let's see the catalyst used here. The catalyst used here is basically, in case of RNA, catalyst is charged, tRNA is charged, charging of tRNA, and basically ribosome will act as a catalyst. Ribosome acts as a catalyst. The enzyme is a ribozyme, which is 23-sRNA in bacteria. Okay, fine. This is with the translation. Now, one of the main essential or important topics, regulation of gene expression. Basically, we know that DNA carries codons. We know that DNA carries codons which undergo transcription, and this transcription leads to translation. But how we know which DNA undergoes, there are many cells, and in many of these cells 2-meter long DNA is present in each of these cells. Then how we know that which segment of DNA in the trillions of cells in our body will need to undergo this process of transcription to produce an mRNA, to produce a translation? How we know this gene expression is regulated? That is a topic here. We will see some operand models. Okay, fine. Regulation of a gene expression. A gene expression refers to very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, gene expression expressing a gene, which we define in case of cistrons. Cistrons are basically the DNA segments which can undergo transcription to form an mRNA, and this mRNA will get translated into form of polypeptide. This is a central dogma of, think it is central dogma of biology. No, I think it is a central dogma of genetics, or I'm not too sure. Given by Francis Crick. Francis Crick or those two guys did many things. Basically, they gave the double helix model. They gave the central dogma here, which is called central dogma in molecular biology. not genetics, molecular biology, central dogma in molecular biology. And they gave basically about not search of genetic material, semi-conservative nature of DNA. Even though it was not proved by them, it was given by Watson and Francis Crick. And now Francis Crick again gave about this one, which is basically about genetic code. Genetic code. Francis Crick gave about genetic code, that it was a mechanism of genetic code, that there was an adapter molecule. And now actually I think that's all, right? Yes. Regulation of a gene expression refers to a very broad term that may occur in various levels, considering that gene expression results in the formation of a polypeptide. It can be regulated at several levels. In eukaryotes, the regulation could be exerted at basically this regulation for the DNA segment transcription can be transcription of a polypeptide formation. Polypeptide is formed, the gene is expressed. Polypeptide not formed, gene is not expressed. Or we can say it is expressed as a recessive trait or something like that. This could be regulated, it could be stopped or controlled at many levels. First level is transcriptional level, formation of a primary transcript, mRNA formation. First level we can regulate a gene expression is stopping or controlling the transcription process. Now, processing level. We know that after transcription in eukaryotes, there is an unprocessed heterogeneous RNA. This heterogeneous RNA contains both introns and exons. Introns are completely useless and they do not undergo translation. So the splicing is formed. Splicing happens and introns are removed, and this process of splicing can also be stopped. That is also one way of regulating. Then transport of mRNA from nucleus to the cytoplasm. In nucleus is the site of transcription, and when this RNA is not taken out of the nucleus to the cytoplasm, there also you can regulate gene expression. And at translational level, where we can stop the translation process, which is protein synthesis formation. Even though mRNA is transcribed, even though it is completely spliced, even though it is transported from the nucleus to the cytoplasm, at the protein synthesis level, it is stopped. The genes in a cell are expressed to perform a particular function or a set of function. Okay, the genes in a cell are expressed in a specific kind of cell in a tissue, very similar. Basically, the definition of a tissue is similar kind of cell forming together to form a specific function, and for a specific cell it is only able to perform a particular function or a set of function. Basically, for example, if an enzyme called beta galactosidase is an enzyme, is synthesized by Escherichia coli. It is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. Basically, enzymes are biochemical catalyst, and this is a daily metabolism process where an enzyme is used. An enzyme called beta galactosidase is an enzyme that is synthesized by E. coli. This enzyme is synthesized by Escherichia coli, and this enzyme is used to catalyze the hydrolysis of a disaccharide into monosaccharides which is disaccharide of lactose into galactose and glucose. And after this, the bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for the energy source, they would no longer require the synthesis of the enzyme beta galactosidase. Basically, they're saying that there is a bacteria, and that bacteria is E. coli. And this bacteria, if it is in a lactose medium, lactose is a carbohydrate and or polysaccharide which is used to derive energy formation, and this lactose is converted into galactose and glucose, which are simpler polysaccharides, not polysaccharides, simpler saccharides or monosaccharides, which are easier or simpler molecules which are essential metabolism steps used for energy derivation. In such a case, the bacteria use them as a source of energy. Lactose, if it's in lactose medium, it will convert lactose to glucose and galactose for energy formation. But this process, an enzyme is required. Normally it would take days, or not days, it will take more time without an enzyme. It will take many times the factors of time when compared with and without an enzyme. So it will manufacture an enzyme called beta galactosidase. This enzyme will catalyze this reaction for conversion of lactose into glucose and galactose. As long as the bacteria is around the lactose medium, it will take the lactose, convert it into galactose and glucose and use it for energy. But what if the bacteria was not in a lactose medium? It barely was in contact with lactose. Will there be a use for beta galactosidase enzyme? No, of course not. If there is no lactose, there is no beta galactosidase enzymes use, because there is no main reactant itself. In such a case, there would be no longer require then the bacteria doesn't need to synthesize this particular enzyme. There is no need at all. So there will be no longer require the synthesis of the enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environment conditions that regulate the expression of genes. Important statement: it is the metabolic, physiological or environmental conditions that regulate the expression of genes. Because of the environment, where E. coli, if in lactose, it was in the lactose environment, it needs this enzyme for the production of energy. But if it's not in a lactose surrounded enzyme environment, there is no need for the enzyme, so it should be regulated. So we can say it is a metabolic, physiological or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organism are also a resulted coordination regulation of expression of several set of genes. This can also be remembered. And development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. Many sets of genes are coordinated and regulated, then only a development from embryo to organism is seen. In prokaryotes, control of the rate of transcriptional initiation is a predominant site for the control of gene expression. What does that mean? Basically, it means in prokaryotes, control of the transcriptional initiation is the predominant site for control of gene expression. An important statement. Basically, it means in prokaryotes, the rate of transcription, basically we saw in prokaryotes, transcription and translation happen at the same time, and transcription is not completely happened, only it's in halfway also. Translation will start, protein synthesis will start. So we can say, we saw a statement that's saying that in prokaryotes itself we can see easy expression of the physiological expression, because it can easily provide new protein. So this protein will be easily expressed. This is due to lack of distance between the cytosol and the nucleus regions. In prokaryotes, this control of because of such a reason, because of transcription acting as because directly from transcription we can directly initiate translation as well in the midway and directly get the protein which will get easily expressed. So in such a case, the first step, transcription, this initiation of transcription is the main factor or a site for control of gene expression. Is a control of gene expression, site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize start sites. Okay, first statement, first information, they are saying that transcriptional site in case of prokaryotes is a main controlling agent or controlling site, that is controlling the gene expression. In a transcription unit, promoter, structure and terminator, the activity of RNA polymerase at a given promoter in turn regulated by interaction with accessory proteins, which affects its ability to recognize the start sites. We know that the transcription doesn't start from any place at all. There are certain specific places called the origin sites, origin of replication sites. Only in that places the RNA will start to segment and it will start to unwind and the process will start. So basically, for the transcription to happen, the transcription unit, the activity of RNA polymerase, it should be connected to a given promoter region, and this as we need to control the gene expression, as we can inhibit the RNA polymerase from binding to the origin of replication site, we can stop we can control or regulate the gene expression. That is what they're saying here. And this is done by accessory proteins, which affect the RNA polymerase ability to recognize the start sites. Accessory proteins which kind of cover the origin sites and will make the RNA to be unable to recognize the start site. In such a case, and we don't need to cover the whole RNA or DNA. This happens, as I said before, due to origin of replication sites. This is also the reason why vectors are used, and vectors can independently divide and they provide origin of replication sites. These regulatory proteins, okay, let's continue here. These regulatory proteins, accessory proteins, can act both positively. This can act as, they can guide the RNA polymerase to push it towards the start of replication site and initiate and catalyze the reaction, not catalyze actually. And we saw actually there was a sigma and rho factors which helped in the initiation. RNA polymerase only acts as a catalyst in case of elongation. The initiation and termination phases in case of prokaryotes were not by RNA polymerase. It was by other factors, which were the sigma and the rho factors. And these regulatory proteins can act as positively activators and negatively repressors or inhibitors. The accessibility of promoter region of prokaryotic DNA in many cases regulated by the interaction of proteins with sequences termed as operators. Basically, these proteins, because they are controlling the RNA polymerase meeting with the start site and starting the initiation, they are called as operators. These proteins are called as operators. The operator region is adjacent to the promoter elements in most operons, and in most cases the sequence of the operator bind a repressor protein. Basically, the operator region, which is basically the interaction of proteins, which will control the interaction of the RNA polymerase with the promoter regions. These are operators, and the operator region is adjacent, side by side, with the promoter elements in most operons, because it is controlling the promoter. Initiation is what needs to be controlled. So these operators, they should be present near the initiator or promotion sites itself, the promotion unit of the transcription unit. There only it should be present normally. And in operons, in most operons, and in case this structure itself, this model is called as an operon, operator, operon, and in most cases the sequence of the operator bind a repressor protein. Okay, their adjacent operator are adjacent to the promoters. Fine. The next information they gave is the sequence of the operator bind a repressor protein. Okay, they contain a repressor protein, basically. I think that's what it means. Each operon has its specific operator, and a specific repressor. For example, we are going to study about the lag operon or operator is present. This lag operator is present only in the lag operon, and it specifically interacts with lag repressor only. Lag operator is present only in the lag operon, and it interacts with the lag repressor bind protein only. Now let's see the lag operon. The elucidation of the lag operon was also a result of close association between a geneticist, Francois Jacob, and a biochemist, Jack Monod. How they discovered they elucidated the lag operon was through an association between a geneticist, Francois Jacob, and a biochemist, Jake Monod. They were the first to elucidate a transcriptionally regulated system. In lag operon, here lag refers to lactose, polycystonic structural gene is regulated by a common promoter and regulatory genes. Polycystonic meaning a single RNA or a single transcription unit is going to produce an mRNA in such a way that this mRNA can code for many kind of proteins. It can express many kinds of proteins with only one kind of transcription unit. This is called a polycystonic structural gene, and this structural gene, this is prokaryotes, it is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as an operon. Basically, this lactose producing or polystructural gene, not lactose producing, sorry, it is not lactose producing, it is a polycystonic structural gene which is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as an operon. To name a few examples, lag operon, TRP operon, ARA or ARA operon, HIS operon or HIS operon, VAL or VAL operon, etc. The lag operon consists of one regulatory gene, that is the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor. Here regulatory gene is an inhibitor gene, and three structural genes Z, Y, and A. The I gene codes for repressor of the lag operon. Okay, these genes code for the repressor of the lag operon. The Z gene codes for beta galactosidase. If you might get confused at why each of these genes is under a single transcription unit is coding for many kind of proteins, that is the beta galactosidase, the repressor enzyme, the repressor gene or repressor protein. Basically, it's because it's a prokaryote and it's a polycystonic unit. It can code for many proteins, polypeptides. So the regulatory gene I gene here does not, and this codes for this I gene codes for a repressor protein, a regulatory protein. And there are three structural genes which are Z, Y, and A. which constitute the main lac operon. Actually, even the I gene contains the main operon itself, but basically the main function of the gene. Instead of acting as an accessory gene for the main gene, which is the function of the I gene, which is actually only acting as a regulation gene for the gene, like for the DNA, it's acting as a regulator. It is controlling the DNA itself or gene itself, gene expression. But here the main gene expression is derived from the other main structural genes. The Z gene codes for beta galactosidase, the beta gal, which is primarily responsible for, we saw this before in case of Escherichia coli, we saw this here itself, is primarily responsible for hydrolysis of disaccharide lactose into monomeric units, galactose and glucose. The Y gene codes for permease, which increases permeability of the cell to beta galactosides. The A gene codes for a transacetylase. Hence, all the three genes and basically the three gene products in lac operon are required for the metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Basically, there are four kind of genes here. Four kind of genes in the lac operon. The lac operon consists of the I gene, which is the inhibitor gene, which codes for the repressor protein, polypeptide, which will repress the or reduce the, which will basically control the gene expression. And there is Z, Y and A, where Z gene codes for the main enzyme, the beta galactosidase, the beta gal, which is primarily responsible for the hydrolysis of disaccharide lactose into monomeric units of glucose and galactose. The Y gene codes for a permease, which increases the cell's permeability to beta gal, and the A gene codes for a transacetylase. Hence, all three gene products in lac operon, basically all these are required when we want the metabolism of lactose. The cell is in the lactose environment and it wants to derive energy from lactose. All these three genes are required for the proper metabolism of the lactose. And in case of other operons as well, the genes present in the operon needed to function together in the related metabolic pathway. This is the basic schematic representation of the lac operon here. Schematic representation of the lac operon. In absence of an inducer, this is the repressor. Repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. Basically, if there is no inducer, then this repressor will bind to the origin of replication site, preventing the the RNA polymerase to bind there and start the transcription process. The repressor mRNA is coded by the inhibitor gene. This is the repressor mRNA, and this will code the repressor protein, and this repressor protein will be binding to the transcription site. And in presence of an inducer, then what happens is repressor mRNA will get the protein is produced, but before binding to the origin of replication site, here there is an inactive due to an inducer which might possibly be lactose. I think that is the case in case of a lac operon. In a lac operon, is the inducer lactogen? I mean, not lactogen, lactose. I think that is the case. It is not lactose actually. Okay, here it's how lactose enters the cell via permease Basically, this lactose we saw here, the Y gene codes for the permease, which increases permeability of the cell because it is in present of the lactose enzyme. If it cannot take the it's not an enzyme, not lactose enzyme, lactose polysaccharide. This lactose polysaccharide should enter the cell. If it can't enter, then energy derivation is not possible. So Y gene will code for that. Lactose enters the bacteria cell via permeases, which is coded by the Y gene. A small amount of this lactose is converted into allolactose by the enzyme beta galactidase, encoded by the LAC-Z gene. Allolactose binds to the LAC repressor protein encoded by the LAC gene, causing it to change shape and detach from the operator region. Basically, how this knows how this inducer is produced is because this is in the presence of lactose only. In the presence of lactose, lactose is converted into allolactose, and this allolactose acts as an inducer. It will bind to the repressor and make it inactive. And if this repressor is inactive, there is no binding at the site of origin of replication, and there is these genes will continue to RNA polymerase will bind here, and there will be a transcription process. The LAC mRNA is formed. It will lead to process of formation of the enzyme beta galactosidase, permease, and transacetylase. Lactose is the substrate for the enzyme beta galactosidase, and it regulates switching on and off the enzyme. Hence, it is termed as an inducer. Yes, lactose is the substrate, but more detailedly, it is converted into allolactase. But in case of NCERT, in terms of NCERT, it is lactose itself is the inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of bacteria, the lactose is transported into the cells through the action of permease. Permease, remember, a very low of expression of lactoferrin has to be present in the cell at all times, otherwise lactose cannot enter the cell. Basically, they are saying that if permease itself is not present, then how can the lactose, which is the inducer, can enter the cell itself? Not enter the cell if there is no lactose, if there is no permease enzyme, the lactose itself cannot enter the cell, and it cannot even act as an inducer because of the lack of the enzyme, the permease. So for it to enter the cell, for it to gain the ability to enter the cell, there should be always a low expression of this genotype. It should be somewhat expressed. Some of the permease beta galactosidase, basically if there is no beta galactosidase produced, then how will the lactose get converted into allolactose? There is no chance to act as an inducer. So there should be a low expression at very low level of the LAC operon. It should be present in the cell at all time. Otherwise, lactose cannot even enter the cell. The lactose then induces in the following manner, operon in the following manner. The repressor of the operon is synthesized all time. Basically, the genes which will be limited will only be the ZYA. The inducer gene, inhibitor I gene is always producing a repressor RNA. It is always there. Otherwise, how can it regulate the gene expression? The repressor of the operon is synthesized all time consecutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. Basically, they are saying the repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. That's what I said before. The repressor will bind at the origin of replication site and prevent the RNA polymerase. The presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. When at some time due to concentration of lactose, it is acting as an inducer and reduce the interaction of the repressor, then the origin of the replication site will continue to lose the repressor protein, and it will allow the RNA polymerase to get binded there and start with the transcription process. This allows RNA polymerase access the promoter and transcription process. Essentially, the regulation of lac operon can also be visualized as a regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for the lac operon. Can you think for how long the lac operon will be expressed in the presence of lactose? How long? I think as long as lactose is there, the lac operon will always be expressed, because lactose is always getting converted into the enzyme. Is always getting converted into allolactose and acting as an inducer, completely stopping the repressor. So as long as there is lactose, the lac operon will continue to express its gene, will continue to express physiologically. When there is loss of lactose or less amount of lactose, then the lactose acting as an inducer also will be reduced, and the repressor will then bind to the origin of replication site or the start site. Then it will stop the replication process. Not replication, it is the transcription process. Regulation of lac operon by repressor is referred to as negative regulation. Lac operon is under the control of positive regulation as well, but it is beyond the scope of discussion at this level. Basically, they are saying the regulation of lac operon, that is by the repressor, it is called a negative regulation. By acting as a repressor and stopping the mRNA synthesis, it is acting as a negative regulation. And there is also a positive regulation, but we will not going to discuss in our chapter or at this level. One of the next main topics is the Human Genome Project, which is also referred to as HGP as the abbreviation, is a Human Genome Project. In the preceding sections, you have learned that in the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. Makeup means creation. The genetic creation of an organism or an individual lies in DNA sequences, which is extremely obvious, because we call DNA as the blueprint of us or a cell. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequences human genome was launched in the year 1990. This Human Genome Project completed. Yeah, they completed it. Started 1990, initial completion announced around April 2003, 50 years of after Watson and Crick's DNA structure discovery. Declared 99% complete with 99.99% accuracy at that time. Although the project was considered complete in 2003, some regions of genome remained unresolved, especially highly repetitive and hard to sequence regions like the centromeres and telomeres. So what's new? In 2022, a group called the telomere to telomere T2T consortium published the first in 2022. It was kind of recent. First truly complete human genome sequence, filling in all the missing part, about 8% of the genome that was left unresolved by HGP, which is the Human Genome Project. Basically, it took 13 years for the HGP to give an approximate 99% of the genome project, human genome. Okay, let's see. Sequencing basis, let's read the paragraph again. DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques which were possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. You can imagine the magnitude of the requirement for the project. We simply define the aims as the project follows of the project as follows. Human genome is said to have approximately 3 to 10 power 9 base pairs, and the cost of sequencing requires $3 per base pair. The estimated cost in the beginning for sequencing $3 is for one single base pair. That is, for single base pair we needed $3. That is in 1990. The total cost estimated for the project would be approximately $9 billion. Further, if obtained, sequences were to be stored in typed forms in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. Very, very large, because there are 3 to 10 power 9 base pairs. We need first problem is the money $3 per base pair, $3 billion. That is at 1990. That much money was required, or at the beginning. And storage, where should we store this many letters? 3 into 10 power 9 base pairs. Where can we store that many amount of letters? We need 3,300,000 page books to store such letter. That too, congesting 1000 letters in a single page. We normally have 100 to 200 words or letters. Wait. Let's leave it be. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development for a new area in biology called bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows. Identify all the approximately 20 to 25,000 genes in human DNA. Determine the sequences of the 3 billion chemical base pairs that make up human DNA. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sectors such as industries. Address the ethical, legal, and social issues that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and National Institute of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, and the Azores. The project was completed in 2003, knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent thousands of diseases or disorders that affect the human beings. Besides providing clues to the understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their chemical capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation, and others. Many non-human model organisms such as bacteria, yeast, cyanor, Habitus elegans. Let's see how to pronounce that. A free living non pathogenic nematode. Basically, it's of the nematoda class, or basically the coelenterata class, not class, I mean phylum. Pronouns. Caenorhabditis elegans. Caenorhabditis elegans. Fine. Caenorhabditis elegans is a free living non-pathogenic nematode. Drosophila, the fruit fly, Drosophila melanogaster, the fruit fly, plant such as Oryza sativa, that is rice, and Arabidopsis, etc., have also been sequenced. Methodologies. The methods involve two major approaches. The method of sequencing, how we can sequence. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequences, and later assigning different regions in the sequence with function, a term referred to as sequence annotation. Basically, you have two methods, two major approaches for the sequencing, genome sequencing. First is identifying all the genes that are expressed as expressed sequencing tags and finding the sequences in that separately or individually. First, finding the genes expressed and naming them as expressed sequence tags. Only the expressed sequence tags are found out, and sequences are then found out separately. And the second method, which is the blind approach, is basically start to the end of a human DNA which we sequence blindly without seeing anything. We just see the base pairs A, T, G, C, 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 G, whatever it might be. We just go on naming them, giving them names, and later, after some part of it is completed, we assign specific enzymes or gene expressions to a certain sequence in that DNA. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller size, because DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA. Basically, they are saying for sequencing a DNA from a cell, this first DNA is completely isolated and then is converted into many small pieces or fragments and then identified basically like that. And they are cloned in suitable host using specialized vectors. They are basically cloned in suitable hosts using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease The commonly used hosts were bacteria and yeast, and the vectors were called as bacterial artificial chromosomes and yeast artificial chromosomes. Basically, they will take the cell, they will take the individual DNA from a single cell, they will isolate it, they will separate it into many fragments, and then these fragments, these individual fragments, they will associate it to a vector. Vector will study about more about it in the biotechnology chapter, and with this vector we will associate it, and vector will help providing the origin of replication site, and it will help increase the number of the DNA from, and this is done in a bacteria, which can be bacterial artificial chromosomes, we can call it the vector name, or in case of if it is done in yeast, it is called as yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers that worked on principle of a method developed by Frederick Sanger. Remember that Sanger is also credited for developing method for determination of amino acid sequences in proteins. Sanger basically gave two things. One, he gave that were fragments sequenced by, basically he developed what kind of sequences, like finding out the what kind of sequences will be there in an amino acid. That one he gave. Second one, he also gave developing a method for determining the amino acid sequences in a protein. Both amino acids as well as the sequences, Sanger only gave. Frederick Sanger method. These sequences are arranged based on some overlapping regions present in them. They are based on arranged in overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible because the fragments is in micrometers, our hands are in centimeters. There is no way we can individually pick each fragment and align them. So therefore a specialized computer based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of the chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosome, 22 autosomes and X and Y to be sequenced. It took till 2006 for the 24 chromosomes to be sequenced. Basically, our notion is basically the blind sequencing. Basically, let's read the paragraph again. These sequences, the fragments were sequenced using automated DNA sequencers that worked on a principle of method developed by Frederick Sanger. Sanger is credited for developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping sequence for fragments for sequencing. Alignment of these sequences was not humanly possible. Specialized computer-based programs were developed. These sequences were subsequently annotated, were assigned to each chromosome. Basically, individually they found out and assigned to each chromosome. This is the representative diagram of the Human Genome Project. They will take a cell from the human, they will take the DNA, isolate the DNA, they will separate it into fragments, and they will send it into automated DNA sequencers where the computer will give us the code. And after sequencing the overlapping regions, we will assign it to each chromosome. And the sequence of chromosome 1 was completed in May 2006. This was the last of the 24 chromosomes, 22 autosomes and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. Basically, another challenging task was assigning the genetic and physical maps on the genome. Okay. This was generated using information on the polymorphism of the restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the applications of polymorphism in repetitive DNA sequences shall be explained in the next section of DNA fingerprinting. Okay, fine. Basically what they're saying, challenging task was assigning the genetic and physical maps of the genome. I didn't get what that means. Let's see. What does the above statement from NCERT about regarding Human Genome Project mean? Another challenging task was assigning locating where exactly each gene, assigning genetic and physical maps on the genome, locating where exactly each gene or DNA segment lies on the chromosome. Okay, genetic map based on how two genes are inherited together. This is the recombination frequency. Physical map, recombination frequency, we read in the before chapter, recombination and linkage. Higher the linkage, lower the recombination. Higher the recombination, lesser the linkage. This is between two genes. And physical maps show the exact physical location in base pairs of the genes. Or genetic maps is basically how these two genes are inherited together. And physical map is which base pairs is exactly doing the work in showing the genotypic expression. This step is like creating a detailed roadmap of the entire genome, show where each genes are located using information of polymorphism of restriction endonuclease recognition sites. Okay, restriction REs, okay, restriction enzymes or restriction endonucleases cut DNA at specific sequences. Polymorphism here refers to variation in DNA. This restriction endonucleases we are going to study more about in biotechnology. These are specific, they are DNA scissors which cut the DNA at specific places. Polymorphism here refers to variation in DNA sequences between individuals that affect where these enzymes are cut. What does that mean? Polymorphism here refers to variation in DNA sequence. The variation in DNA sequence between individuals that affect these variants are called restriction fragment length polymorphism. Basically, understanding where the enzyme is cut, how it is affecting, I think they are finding where the gene is actually located. Basically, if they cut an enzyme somewhere, not a DNA somewhere, and this is deleted, and if it how it's affecting the organism, they are going to find out, and based on that, they are going to find the gene location. And some repetitive DNA sequences known as microsatellites. Microsatellites are short repetitive sequences of 1 to 6 base pairs like CA, 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 CA scattered throughout the genome. There are highly variable between individuals, making them genetic markers, useful as genetic markers, because they are very variable in organism or individuals. They help in making useful genetic markers. Same as that, think of microsatellites as landmarks that appear in many places, helping scientists anchor different parts of the genome. Summary, this sentence means that one tough part of the human genome project was figuring out exactly where each gene is located in the DNA, and scientists used variation in enzyme cut sites and microsatellites, repetitive variable DNA sequences, as markers to build a genetic and physical map of the entire genome, the human genome. Now, the salient features of the human genome project or the human genome. Salient features of the human genome. Some of the salient observations drawn from the human genome project are as follows. The human genome contains 3.1647 billion base pairs. 3.1647 billion base pairs. The average gene consists of 3000 bases. A single gene consists about 3000 bases, where sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. The total number of genes is estimated to be at 30,000, much lower than previous estimates of 80,000 to 1,040,000. I mean, not 1 million, 1.4 lakh genes. Almost all 99% nucleotide bases are exactly the same in all humans. 99% of all the nucleotide bases are exactly the same in all humans. This is why we are a single species. The functions are known for over 50% of the discovered genes. Basically, the functions of 50% of over the 50% of the discovered genes are unknown. They don't know what these genes do. Over 50% of the genes. Less than 2% of the genome codes for proteins. Repeated sequences make a very large portion of the human genome. Repeated sequences make a very large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes hundreds of thousands of times. They are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has the most genes, 2968, and the Y has the chromosome Y has the least, which is 231. Scientists the chromosome 1 has almost 13 to 14 times the genes, or 12 to 14 times the genes of the Y chromosome. Scientists have identified about 1.4 locations where single base DNA differences, single nucleotide polymorphism, pronounced as SNPs, occur in. What is nucleotide polymorphism? Polymorphism, variation in DNA. Okay, polymorphism. Have identified about 1.4 million locations where single base DNA differences, SNPs, occur in human. Basically, where the single insertion occurs. Basically, there are 1.4 locations where single base is different for different human beings, and it affects the genome expression. This information promises to revolutionize the process of finding chromosomal locations for disease-associated sequences and tracing human history. Now, one of the last topics, not last topics, I think DNA fingerprinting is the last topic. Yes, DNA fingerprinting is the last topic. But before DNA fingerprinting, we are going to see the application and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and the private sectors of the worldwide. Deriving meaningful knowledge from whatever the Human Genome Project or a DNA sequence will define research to the coming decades through the tens of years, leading to our understanding of the biological system. This is a very, very huge task and will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having the HG sequence, human genome sequence, may be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time. When we think about seeing something, when we know DNA is a blueprint, normally it is kind of obvious for us to first focus on the actual DNA to see the problems or disorders in a human, but that is actually not how it is done, because we didn't know in the previous years we didn't know how the DNA actually is, what is even the DNA, we didn't know, but there was doctors at the time. So the obvious approach, which was seeing the DNA in the first place, became the hardest and was not actually followed. But now when we have the human genome, the whole human genome sequence, we can be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time, when a whole genome sequences and new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. All the transcripts in a particular tissue or organ or a tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. Once more, the chemistry of life. Now the final topic under the molecular basis of inheritance is DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. Assuming human genome as 3 into 10 power 9 base pairs, in how many base sequence would there be a difference? Basically 99% is the difference. So it should be approximately a difference with 10 power 3 into 10 power 9 by 100. 3 into close to 100, 99.9% is close to 100. So 3 into 10 power 9 by 100, it is 3 into 10 power 7 base pairs. This many base pairs, which is 30 million base pairs, we see a difference. Even though we say 99.9% is same, we see a difference in case of 3 into 10 power minus 7 base pair, which is a very, very large number. It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. Because we are varying with a very, very large 3 into 10 power 7 base pair number, we change in a phenotypic appearance as well. If one aims to find out genetic differences between two individuals or among two individuals for population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 power 6 base pairs. Wait, not 7, it is 6 base pairs, which is for one base pair it is $3, that is at few decades back. Now the cost is going to be even more, and the DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific sequences called as a repetitive DNA. This is the microsatellite DNA, where this is very varying within individuals. Because in these sequences a small stretch of DNA is repeated many times, these repetitive DNAs are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak, and the other small peaks are referred to as satellite DNA. Depending on the base composition, A and T rich or G and C rich, length of segment and number of repetitive units, the satellite DNA is classified into many categories such as micro, mini, etc. These sequences normally do not code for any proteins, but they form a large protein portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA form every tissue such as blood, hair, follicles, skin, bone, saliva, sperm, etc. From an individual, the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequences, the basis of genetic mapping of human genome as well as the DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism, variation at genetic level, arises due to mutations. Recall different kinds of mutations and their effects that you have already studied in chapter 4 and in preceding sections in this chapter. New mutations may arise in the individual, either in some somatic cells or in the germ cells that generate gametes in the sexual reproducing organisms. If a germ cell mutation does not seriously impair the individual's ability to have offspring who can transmit the mutation, it can spread to the other members of the population through sexual reproduction. Allelic, again, recalling the definition of allele, sequence variation has traditionally been described as DNA polymorphism if more than one variant allele at locus occurs in human population with a frequency greater than 0.01. In simple term, inheritable mutation is observed in a population at high frequency. It is referred to as DNA polymorphism. If an inheritable mutation is observed in a population that is at a high frequency greater than 0.01, it is referred to as a DNA polymorphism. The probability of such a variation to be observed in non-coding sequences is higher, as mutations in these sequences may not have any immediate impact in an individual's reproductive ability, because these not affecting the reproductive ability. So if it is affecting the reproductive ability, it will not be heritable. If it is not affecting the reproductive ability, it will not be it will be heritable. So these changes will continue to spread. These accumulations keep on accumulating generation after generation and form one of the basis of variability polymorphism. There is a variety of different types of polymorphism, ranging from single nucleotide change to very large scale changes. For evolution and speciation, such polymorphism play a very important role, and you will study these details in details at higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as probe that shows a very high degree of polymorphism. It was called a variable number of tandem repeats, V and TR. The technique, as used earlier, involved southern blot hybridization
using a radio labeled VNTR as a probe. It included isolation of a DNA, first step, digestion of a DNA by restriction endonucleases. Basically, this is cutting off the DNA into many fragments. Separation of the DNA fragments based on the density gradient through centrifugation. No, wait, not by centrifugation, by electrophoresis. We know that our DNA is negatively charged, and if we place it in a plate, here, basically in this separation by size on a gel, basically gel electrophoresis, basically because based on the mass they will get separated as due to attraction forces from the negative charges when we place it in an electric field, transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, hybridization using VNTR probe, and direction of hybridized DNA fragments by autoradiography. A schematic representation of the DNA fingerprinting is shown in figure 5.16. Schematic representation of DNA's fingerprinting. A few representative chromosomes have been shown to contain different copy of VNTR. For the sake of understanding different color schemes have been used to trace the origin of each band in a gel. The two alleles, parental and maternal, of a chromosome also contain different copy of number of VNTR. It's clear that banding pattern of the DNA from crime scene matches with individual B and not A. This is the DNA from individual A and DNA from individual B. If we see the sequence, there are two red strands here, two yellow strands here, two blue strands here. And between the two individuals, when you see the DNA sequence, B and C are exactly the same. So the individual B is the criminal. The VNTR, which is basically variable number of tandem repeats, belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy numbers varies from chromosome to chromosome. In an individual, the number of repeats shows very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with the VNTR probe, the autoradiogram is basically, there will be DNA in the gel electrophoresis. If you see, there will be DNA present on the sheet here, but how we should observe them? DNA are very microscopic and they are not colored, so we cannot observe them easily. So we use the autoradiogram to identify them. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population, except in these cases of monozygotic twins, basically twins formed from a single zygote. The sensitivity of the technique has been increased by the use of polymerase chain reaction (PCR). This is in biotechnology. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider application, such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. I am indeed going to read the summary. Nucleic acids are long polymers of the nucleotides, while DNA stores genetic information. While DNA stores the genetic information, RNA mostly helps transfer and expression of information. Though DNA and RNA both function as enzyme and genetic material, RNA being chemically and structurally more stable is a better genetic material. However, RNA is the first to evolve and DNA was derived from RNA. This is the RNA world. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases of the opposite strands. The Chargaff rule basically saying that the number of purines is equal to the number of pyrimidines. The rule is that adenine pairs with thymine through 2 hydrogen bonds and guanine with cytosine to 3 hydrogen bonds. This makes one strand
complementary to each other. The and this is also the reason why the distance between each the distance in the DNA helix is constant. The DNA replicates semi-conservatively. The process is guided by complementary hydrogen bonding. A segment of DNA that codes for RNA may, in a simplistic term, can be referred to as template to direct the synthesis of the complementary RNA. In a bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split, the coding sequences, exons are interrupted by non-coding sequences, introns. Introns are removed and exons are joined. And the introns are removed and exons are joined or shuffled to produce a specific RNA by splicing. The messenger RNA contains the base sequences that read in a combination of 3 to make triplet genetic code to code for amino acid. The genetic code is read again on the principle of complementarity by tRNA that acts as an adapter molecule. There are specific tRNAs for every amino acid. The tRNA binds to each specific amino acid at one end and pairs through hydrogen bonding with codes on mRNA through its anticodons. The site of translation of protein synthesis is ribosomes, which bind to mRNA and provide platform for joining amino acids. One of the RNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme ribozyme. This is in the prokaryotes. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive processes, these have to be tightly regulated. Regulation of transcription is the primary step for regulation of the gene expression. In bacteria, that is, more than one gene is arranged together and regulated in units called as operons. Lac operon is the prototype operon in bacteria, which codes for genes responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project was a mega project that aimed to sequence every base pair in human genome. This project was yielded much new information. Many new areas and avenues have opened up as a consequence of the project. DNA fingerprinting is a technique to find out variations in an individual of a population at the DNA level. It works on the principle of polymorphism in DNA sequences. It has immense application in the field of forensic science, genetic biodiversity, and evolutionary biology. With this, we have completed first revision. Of molecular inheritance of biology. Via NCERT. That is completed. Okay then, see you in the next video.